First question, what is plasma? If you Google plasma on the internet, you'll probably see this image right here. These are all the examples of both man-made and plasmas found in nature. It doesn't really tell you a whole lot about what plasma actually is. The nicest and briefest description for me for plasma is it's a gas that can conduct electricity. And that's it. That's what plasma is, a gas that can conduct electricity. I think the most common plasma that you'll encounter in everyday life is a fluorescent tube. That is a glow discharge. One of the byproducts of a plasma is the light that it gives off. So we use plasma to light the darkness. What we use at PVA TePla is the chemistry, which is the byproduct of the plasma. That chemistry we use to change properties of surfaces. We alter those properties to clean, promote adhesion. Okay, let's talk a little bit more about plasma. Plasma, you'll hear as well, is the fourth state of matter. If you add energy to a solid, eventually you'll overcome some intermolecular forces, you get a liquid. Keep adding energy to the liquid, you'll overcome more intermolecular forces, you get a gas. Keep adding energy to a gas, you'll eventually start to break away some electrons from the gas particles. Once these electrons break away, your gas suddenly becomes conductive. You've got a plasma. That's the fourth state of matter. Okay, this is really what we do at PVA TePla. We alter the surface chemistry of materials using plasma. So I want to talk a little bit about surface chemistry. On this slide right here, this is everything that plasma can do to modify your surface. It's all on this slide. So these 5 points, surface etching, which includes surface cleaning, plasma surface activation or passivation, this is really raising surface energy or reducing the surface energy. Plasma deposition of different polymers like silicon dioxide or Teflon or diamond-like coatings. This fourth one is a little bit more complex. This fourth one is really chemical grafting. This is grafting different functional chemistries to your surface, like primary amine groups or carboxylic groups, etc. And finally, reduction of, for example, metal oxides. So I'm going to talk about each one of these different applications for plasma. Okay, this is the first one, surface etching. There's two ways that plasma can etch a surface, etch or clean a surface. The first is a chemical etch. This is where we will react plasma particles with the surface to form new volatile compounds that can be removed by the pump if you're using a low pressure plasma. The other method is the physical etch. This is where we will accelerate particles towards the surface, and they will act as a ballistic effect and physically knock particles off your surface. So those are the two main mechanisms for surface etching or surface cleaning. Surface activation or passivation, this is where we raise the surface energy or we reduce the surface energy of a material. Raising the surface energy, this is where we make energy available to bond with an adhesive or an ink. At surface passivation, this is where we make the surface inert. One of the ways that we can do that, for example, is by adding fluorine groups or coating with a Teflon-like coating. And that brings me on to this slide, surface coating. There's a number of different things that we can do with surface coatings. We can either deposit functional coatings or chemical coatings. Now this really is a plasma-enhanced chemical vapor deposition or PECVD process. The functional coatings you can see here, they range from PTFE, quartz-like coatings, diamond-like. We can deposit polyethylene glycols or polyethylene oxide-like coatings using PECVD, and also hydroxyethyl methacrylates. We can also deposit coatings that contain a chemical functionality. So let's say you need a surface that has a primary amine functionality or an epoxide group. We can do that using this process, a PECVD-like process. Okay, this is just to give you an example of an untreated plasma surface. If you're trying to deposit a coating without first treating the surface, you can be sure that there's going to be contamination on the surface of your product. The second thing is because there's no chemistry on the surface, there's no way of predicting how your functional molecule or biomolecule or whatever coating you're trying to get down on the surface, there's no way of predicting how it's going to bind. It might bind active end down, in which case you've lost your functionality. So the first thing that plasma will do, it removes the contamination, it cleans your surface, it normalizes your surface. The second step that plasma can do is actually put down, I've depicted them here as purple cups, but it can deposit a specific chemical functional group. Like I mentioned before, a primary amine or carboxylic acid or an epoxide, something like that. So then when you do expose it to your functional molecule, let's say if it's aldehyde terminated or carboxylic terminated, and these are primary amines, you know it's going to go aldehyde end down. And then oxide reduction, just what the picture tells you. It's removing the oxide from a metal. I wish we could do it as good as this, but these are XPS graphs of, this is copper, and this is nickel. Basically what we're showing you here is that as a function of time, we can increase the signal from pure copper and reduce the signal from the various different copper oxides on the surface. This is using hydrogen-based chemistries. We're looking here at nickel. Nickel is sort of an odd one. You do see an initial increase in the nickel oxide concentration, followed by a sharp reduction. It does take a little bit longer, but we can do it. We have demonstrated that we can reduce nickel oxides as well. Okay, let's talk a little bit about the chemical functionalities that we're able to get down on surfaces. You can see them across the top here: amines, hydroxyl groups, carboxyls, aldehydes, epoxides, and sulfhydryls. And I went to the literature and I found all of these different types of biomaterials that were deposited on the surface using these functionalities. So, for example, there's a lot of publications on the adsorption of cells to surfaces using amine groups in particular, but also hydroxyls and carboxyls, oligonucleotides and DNAs and proteins and enzymes. A lot of papers using epoxide functionalities. Okay, there are two main ways of plasma treating a surface, trying to chemically functionalize the surface. The first is a straight activation process, and the second is what we call a plasma polymerization process. This is by far the better method. A plasma activation, let's say you're looking for a primary amine group. So you feed ammonia gas into your chamber. You will get about 2% primary amine on your surface using ammonia gas. On the other hand, if you feed in some monomer species that you can polymerize on the surface that contains that functional group, for example, an allylamine, you will get in excess of 10% primary amine group on the surface. So it's a lot easier to control the chemistry if you use this method here, but both methods are possible. Okay, let's talk a little bit about some of the applications. A lot of the applications that come through our facility is for cleaning, precision cleaning of parts. And again, we talked about it in the introduction. There are two ways to clean the surface. Let's say if you have a hydrocarbon contaminant on your surface and you want to remove it. Probably the best precursor gas would be oxygen. So you feed oxygen into your chamber, you activate it with the plasma. This oxygen will convert that organic contaminant into CO2 and water. The exact same combustion process as what happens when you burn gasoline in your car. And those are the products that are going out your pump. The other method is physical method. Let's say you had an inorganic compound that's very difficult to remove by converting it into some kind of volatile chemical. You have to remove it physically by accelerating ions towards the surface and more or less sputtering them off. This slide just shows you some of the examples of materials that we can remove from surfaces. Machining oils, mold release agents, polymer bleeds, drill smear, carbon ash, fingerprints, etc., etc. This is an interesting one here, proteins, DNA, and carbohydrates. Unfortunately, we can't call ourselves a sterilization technique, but plasma does sterilize. It sterilizes a lot better than hydrogen peroxide, because not only do you get your 6-log kill, but you also remove the debris that's left behind, your proteinaceous debris. Adhesion promotion. There's actually 4 ways that we can promote the adhesion of either an ink or an adhesive or some sort of a coating that you're trying to deposit on the surface. The first thing that we talked about already, the plasma will clean the surface. It will remove all the contamination. The second thing that it can do is raise the surface energy. It can make energy available on the surface to bond with your adhesive or your ink or your coating. We can also roughen the surface through bombardment. Now this actually, either through bombardment, we can also use a chemical technique to roughen the surface. So of course the rougher your surface is, the more surface area there is for binding. Is it too loud? And finally, chemical priming. This we talked about already. So these are the 4 ways that we can promote the adhesion of either an ink or an adhesive or some sort of a bio-coating to your surface. This slide here just shows you some of the improvements in adhesion strength for different materials. I can't remember what the adhesive is, to be honest. Everything is in pounds per square inch, and this is the gain ratio. So, we're looking at 6 times improvement for polyimides, 24 times improvements for polysulfones. PTFE, we've got 50 plus times. Actually, that's polyethylene, TEF-cell. PTFE we can do. The standard method for activating PTFE towards adhesion promotion is to use an ammonia plasma. This is the result that you get with an ammonia plasma. You've also got a time limit in which to do your downstream processing. The time limit is about a half a day, which is it's not very long, particularly if we're trying to demonstrate what we can do and we've got to send parts back in the post. So we developed a new method using an alcohol plasma. The adhesion strength improvement is much better. This graph here shows you the contact angle over time. We actually pulled a part out of our lab that we did a year ago. We still have contact angle of less than 20 degrees from a starting point of about 115. Lubricious coatings. So plasma will not actually deposit the lubricious coating. What gas plasma surface modification does is it preps the surface so that you can absorb your lubricious coating to that surface. This is, again, an example. I just pulled it out of the literature. It's an argon oxygen plasma treatment of these IOL, intraocular lens delivery devices. They need to put a lubricious coating on these devices so that when they squeeze the lens into the eye, it goes in easily. It doesn't damage the lens itself. This is the technique that they use. So they needed to clean the surface and activate it so that this lubricious coating would adhere and would not come off when the lens was injected through the cartridge. Hydrophilic surfaces, the exact same principle. Plasma can be used to make surfaces permanently hydrophilic, or we can activate a surface so we can deposit another coating that has hydrophilic properties. Some examples here, these are well plates. There, 384 well plates. What we have here is a bubble. This bubble formed because the surface of this polymer is hydrophobic. After plasma treatment, you never get bubbles when you dispense a fluid into these wells. For the activation of tissue culture media, we were able to improve the adhesion properties of cells to the substrates. We no longer found areas where there were no cell attachment, and we reduced the amount of clumping that we observed without plasma surface treatment. With plasma surface treatment, yeah, we got nice uniform adhesion of cells. Anti-biofouling coatings. Again, it's the same principle. We use plasma to chemically functionalize the surface so that when you expose it to your functional coating, for example, an anti-biofouling coating, that coating will adhere to the surface. In this particular example, this is a peg chain. It's aldehyde terminated. The surface was chemically grafted with primary amine groups. Now, if we got a low density of primary amine groups on the surface, when we expose it to the peg chain, the concentration of the peg was not high enough. So when it was hydrated, it formed this kind of a spiral, which left access to contaminants or proteins or microbes to actually reach the surface. If we get a high enough density on the surface, this doesn't happen. When you hydrate the peg chain, it entangles with each other, forming a nice barrier right here. Antimicrobial coatings, same principle. Again, this is just pulled from the literature. They were required a primary amine functionality and a glutaraldehyde linker to put down their antimicrobial coating. These are just examples. There's 1,001 ways to skin this cat. Antithrombogenic coatings. Again, in this case, they were looking for carboxylic acid functionality on the surface. Tissue engineering. This is an emerging area. These tissue scaffolds are often polylactic acids or polyglycolic acids. To promote the adhesion, particularly for these porous type scaffolds, they use ammonia plasma. Cells don't seem to mind whether it's a primary amine or a secondary amine, so in this case, using a plasma polymer is not critical. Doing a straight ammonia plasma activation seemed to work very, very well. When we compared this technique with a wet chemical activation using ethanol, for the tightly poured scaffolds, we were getting about 80% cell seeding efficiency, whereas with the ammonia plasma treatment, we were still in excess of 99% cell seeding efficiency. Drug delivery coatings. Okay, there are two types of drug delivery coating. One is a coating that goes on top of an antibiotic and allows the drug to slowly release into the surroundings. The second one is for the likes of inhaler devices where you're trying to meter the amount of drug that goes into the inhaler or that's carried by the propellant. In this case, we do a Teflon coating to prevent the drug from adhering to the inner walls of the metering chamber. This is the drug release coating. It's a plasma deposited poly-N-butyl methacrylate. After direct deposition of this polymer, we get kind of a biphasal elution of the drug. If you oxygen plasma treat this coating, kind of cross-link the surface afterwards, you get a nice uniphasal release over 40 days. Friction reduction or surface tack reduction. We can actually reduce the surface tack of materials like nitrile rubbers or silicone rubbers or acrylates using plasma. There's a number of different ways of doing it. We can either use a fluorination technique, or we can deposit coatings. And there are other cross-linking methods that we can also use by bombarding inert gases onto the surface that reduces this tacky nature. Again, we're comparing the reduction of the coefficient of friction using plasma. We're comparing it to other methods like fluorine ion implantation, fluorine gas treatment. This is the plasma right here. We get the lowest mean coefficient of friction after plasma treatment and the lowest standard deviations. This is an interesting slide, actually. This is an SEM of nitrile rubber. You can see the mold lines of this. It was an O-ring that we used for this experiment. These are the molding lines that we observed using SEM. After plasma, these molding lines had completely disappeared, and yet we were not etching this surface at all. So we did some extended plasma processes to see what the etching rate was, because when we saw this, we thought we were actually etching the material. It turns out we weren't. So we're hypothesizing as to how plasma works. What we think is happening is plasma is breaking and activating these long chains at the edges or sharp corners. They're kind of folding down and reabsorbing to the surface is what we think is happening. Okay, I'm just going to run through these slides very quickly since I'm running out of time. These are just some of the applications by market. We've already mentioned assays, chemical assays, culture media, microarrays. We treat consumables, laboratory consumables, like, for example, reservoirs, for maximizing the recovery of reagents at the bottom of these reservoirs. We activate these needle hubs prior to adhesively bonding and potting these needles to the polymer hub. In orthopedics, it's mostly cleaning. After polishing of these hip stems, the chemical polishing, we got to remove the chemical polish. That's done using plasma. Also for PEEK implants for spinal fusion cages, etc. We clean those PEEK parts and activate them as well towards cell adhesion. In the ophthalmics, we make these silicone hydrogels hydrophilic, so that the tear wets across the contact lens and it doesn't irritate the eye. We also do some work in the field of intraocular devices, removing the surface tack from the haptics, for example, so that they unfold readily once they're inserted into the eye. Wounds treatment, we can treat fabrics as well with plasma. And finally, filter membranes. We can treat filter membranes to improve the hydrophilicity and improve wettability. I'll take any questions, and if anybody wants to find out more about plasma, we're in booth 3231.